I'd like to invite Professor Frangi, IEEE Fellow, SPIE Fellow, EAMBS Fellow, MICCAI Fellow, and Professor of Computational and Medicine, University of Leeds, Royal Academy Chair in Emerging Technology, also working for faculties of engineering and medicines, KU Leuven, Belgium, to give his keynote speech on the topic of computational precision imaging and medicine in regulatory science. So please, Professor Frangi.
So thanks a lot. I hope you can hear me. You can just do a thumbs up. That's great. My name is Alex, and I'd like to provide today a talk that is a very high-level overview of the work that I'm doing, funded by the Royal Academy of Engineering. And I think it will show a very interesting use case for AI that I hope will be, will elicit some thoughts and reflections. So the focus of my work is about how we can do better medical devices, and in particular, how can we improve the evidence that we need for regulatory approval. So medical devices are basically everything from implants all the way to digital health solutions, to also AI software, although most of the work that I'm going to present today is really focused on implants. And this is a very important market across the globe, where there is a huge amount of technological convergence, not only from materials and from biochemicals, but also increasingly by the use of AI. And the problem is that as those devices that we are developing become more and more complex, and at the same time, our aging society with a number of lifestyle changes increases our life expectancy, makes the fact that we will need to actually develop devices that will have to be implanted for a longer period of time in our bodies. The problem is that a lot of these devices have led to quite serious adverse effects, and it is estimated that in the period of the last 10 years, about 83,000 deaths have occurred, and about 1.7 million serious adverse effects related to malfunction of medical devices that went through the whole regulatory process. That increases the cost of those devices because the sector has less trust. There is an increased number of regulations that need to be satisfied, and therefore the R&D cost increases as well. And with that, also the delay of bringing new innovative solutions to patients and benefiting patients in the first place. Now, this problem happens throughout the whole life cycle of the medical device products. It's not just in the early innovation phase. And hopefully, I'll show how that can be addressed. Now, the interesting thing is that while this sector, in this plot, I'm showing the increase in demand, both in the pharma and medical devices sector, is about 5 to 6% year-on-year growth, which is similar to the automotive industry. However, if you look at the amount of pharma and medical devices that are coming up through the pipeline of the FDA, so of the regulatory approval, it's flat over the last 20 years. So we have increased demand, but actually a stagnation in the ability to deliver those through the regulatory process. Now, if we look to a very different, but not so big, so much bigger sector, like the automotive sector, is about 4 to 5 times bigger than the medical device sector. And if you see 30 years of development, you show that you can see that there is actually a massive improvement in the products that we can purchase today. But also, and most importantly, in the process by which we build them and we manufacture and engineer them. So we don't tend to do any more wind tunnels. We do that aerodynamic simulations. We also do virtual prototyping, and we are using AI for self-driving cars. Now, for a number of years, I've been puzzled why this is happening in the automotive and aerospace and ship industry. However, in the medical device sector, I show you 30 years worth of development in a medical device that I'm going to talk a bit today, which is the type of devices that are used in brain aneurysms, so enlargements of the vessels of the brain that could burst and produce a hemorrhagic stroke. We see that all the devices over the last 20 years is essentially a bunch of wires that has been, with different slightly changes in the design. Now, they lead to, after 30 years, still to 20% complication rate and 3% mortality rate. And we could utilize the same modeling and simulation technologies that we use for the automotive industry in this sector. So when we talk about regulations, what do we need to prove? Well, we need to prove safety of the device, and we need to prove efficacy of the device. And the traditional pathways to do that is animal experimentation, bench experiments, and humans. What we advocate is the need of in silico or computational methods that actually complement, not fully replace, but complement these other three approaches. So if you see today how the sector is evaluating technologies, is a large proportion of bench testing, then animal testing, then human testing, and very, very little computational models. What we advocate is the need to actually develop much more computational techniques that actually allows us to sift away those medical technologies that through simulation we could already demonstrate that will never be a successful innovation, and therefore reduce the need of bench, animal, and human testing only to those that are strictly essential, and those candidates that are likely to be successful. So in that sense that we can replace, we can reduce, and we can refine the need of conventional trials. So computational medicine is, in this context, the application of scientific methods from engineering, mathematics, and computational sciences to understand the treatment of human diseases. So how does this look in practice, and how this relates to AI? So one way to think about this is that through AI techniques, image processing, computer vision, we could actually develop libraries of anatomical models from very large population databases of medical images. And for it, in this case, what I'm showing is aneurysms, a subset of aneurysms from a very large population that we built. And then what you can do is actually evaluate, insert implants, in this case the stents, in each of those morphologies from this library that you have. And when you do that, you can also look at, for instance, what has been the reduction in flow that has been produced by the presence of the device compared to the situation before the device. The idea being that if we actually reduce the flow or we stagnate the blood, we're actually producing a thrombosis, a coagulation that will essentially reduce the chances for the pressure of the blood flow to burst that aneurysm. Now, do we need only anatomy? No, we also need signals and physiology, and that comes from wearable sensors and from models that we can build about populational variability, the same way that we did populational variability of anatomy. So, for instance, in this case, what I'm showing is, in red is the stress conditions and rest conditions in blue, and the shading around the thick bold line corresponds to the variability across a population. So we can actually assess how for each of these anatomies, different physiological regimes related to patients that, for instance, are sedentary or more sportive would affect. So in this particular case, we have the same device, the same geometry, the different physiological regimes. And of course, you could develop models for different devices and compare them in relation to one another. And what you can see is that the flow reduction, if we assume this is the metric we want to use for success, is very different depending on the different scenarios of testing. One of the problems of the current clinical trials, which is the approach to test devices in humans, is that it's very limited to a very small subset of patients, usually in the order of a few hundreds. And when those devices then are mass implanted in humans, we test their effectiveness in thousands of patients, and therefore everything that can go wrong is likely to go wrong at some point. So how do I see an in silico trial space? It's essentially a very large simulation ensemble where we have anatomical models one side, physiological conditions, it could also be operational conditions, depending on the actual intervention, for instance, and the device design. And we can get data for all of those components from different existing population or industrial-related databases. So from a point of view of AI, the sort of models that I'm talking about are not standard conventional machine learning or deep learning techniques, but actually is a combination of those standard techniques and also more computational sort of modeling and computational engineering pipelines, where we need to go all the way from the original images through a number of processes like segmenting the image, doing surface correction, mesh generation to do finite elements, being able to do, models of the implantation of the devices, setting boundary conditions with models of the physiology, solving those equations, either with finite elements or with, for instance, physics and four-neural networks, and then be able to couple the physics of the flow with the biology of the clot formation process to provide maps of both the flow, circulation, and the concentration of thrombus within the body. Now, if you can do that, not just in manual and sort of artisanal manner, if you could streamline to do this at scale, what you have is effectively a population of virtual twins or digital twins. Again, another topic that comes more and more in the area of data science and AI. And those populations where every column here corresponds to a subject where you have its image, the whole instruction of the anatomy and its phenotyping, and then physics, and then the rest of the data. Each of these vectors allows us either to do diagnostics potentially, but also in the case of the presence of devices, understand the performance of the device within a wider set of data points. So I talked a bit about extracting anatomy, extracting physiology, but we also need to do models of the implantations and the treatments themselves. And here I'm showing an example of the virtual stenting or virtual implantation of the device, that if I would have the time to go in more details, this is essentially models of simplex meshes that are deployed on other surfaces, but actually done in a way that respects some of the knowledge that we have about the physics and the devices themselves. So, and then you can put those devices in the geometries that I talked before and do what we call also in silico interventional studies. I'm not going to go in detail over this one for lack of time, but just to show that these technologies are already being used and we'll be publishing that as well in medical journals. So if I put it all together, what are the main three themes and challenges behind in silico studies, in silico trials, and how they relate to AI and to machine learning? Well, we need to build virtual patients and virtual populations. This is all about generative models, graphical deep learnings, uncertainty quantifications. I could spend quite a bit of time there. It's also about modeling virtual examinations and interventions. So how we implant a device, and as I will mention in a minute, how you do, for instance, virtual imaging technologies. And then finally, and this is all about modeling, simulations, and there is a number of data science, but also more recently AI technologies that incorporate a lot of priors about the domain. And then finally, we also need to do interpretations of those images as if we had an expert that does interpretation, because if we want to model a trial, we need to model every single step. And that is the concept of virtual readers and virtual outcomes, which is essentially predictive models in the case of virtual outcomes, and virtual readers is essentially AI applied to, for instance, imaging in this particular example that I'm showing there. So to show that this is not just implants where this is affected, let me give you just a very quick idea about how this could be applied for imaging. So imagine you have a new imaging technique. This is a virtual examination, is a real examination, and you need to test that device, a new MR scanner or a new modality. And normally the way to do that is you would, make a number of subjects go through that device. You will define some measure of diagnostic accuracy, and then you will have to put a radiologist that does interpretations of those images. So a virtual trial would be to replace those humans with virtual detailed models, virtual twins, digital twins, do the simulation of the physics of the image formation process, and then having some sort of AI that does the virtual interpretation of those images. So sometimes we talk about digital twins representing a single organ, but there are people who are developing quite sophisticated models of the anatomy and also looking at different age groups. So, there is a lot of people working on how to build those. And again, you can imagine computer vision being critical in this area. Sometimes we also need to model not just the anatomy, but also the pathology. So there are people who are developing models that take those virtual models of the anatomies and are able to implant pathology by learning that pathology from real data sets and creating generative models that actually can synthesize those features. And again, I don't have the time, unfortunately, to go in detail, but just to show some of the examples of this. So to finalize, I'd like to show an example in a bit more detail about the use of these in silico trials and how our community is making a difference in in this area, and are starting to feature in some of the most prestigious journals, as AI in general is picking up in the Nature journals, for instance. So one of the things we are working on is in brain aneurysms, and there are 4 types of devices, families of devices, that are very, very useful in this case. Clipping, which requires a craniotomy, so you need to open the skull and put a clip at the neck of the aneurysm. Coils, stents, and flow diversion devices. And perhaps in this image, you can see a bit how the coiling gets inserted into the aneurysms and fills it, so reducing the blood flow and promoting coagulation. Sometimes you can utilize stents that do the same effect, but the problem is that the stents, the conventional coronary stents, are not useful because they have a very broad cell, and they need to be a lot less porous devices to actually redirect the flow. And sometimes there are combinations of stents and coils, especially in wide neck aneurysms, to avoid the coils coming out. By the way, you need to realize that these aneurysms are in the brain, and to get there, you actually go under image guidance from a small incision that is in your groin. So it's everything, completely under fluoroscopic guidance. So what we develop is a bit what I explained before, is, we develop a library of anatomical models where we model different physiological regimes, and we compare a particular device in terms of its quality of closing the aneurysms. We built a pipeline like the one I quickly explained before. We also had to develop a lot of AI algorithms to streamline that processing, because if you do this in the more artisanal way that has been done traditionally with algorithms we have, say, 5 to 10 years ago, this would take about 6 weeks to just process 82 patients' data sets. I mean, when you think that one of these trials can take several years, this is still a small time, but we are streamlining this now so that you can do about every case in about 30 minutes. Okay, so you can scale this up to very large numbers. Just to give a sense about the sort of pipelines that we are talking about, you need not only to do segmentation, as I explained before, but you need to also detect the aneurysms. You need to compute center lines, automatically identify endpoints, and know which are the inflows and the outflows. Then virtually be able to get the neck of the aneurysm, deploy the stent, and then do volumetric mesh generation. And when you think about doing this manually, it doesn't seem particularly impressive, but if you want to do this in a way that there is no manual intervention, this is, I can tell you, quite an endeavor. So we tested all of this in a flow diversion device, in a commercial device from Medtronic. And this device, just to give you an idea, there is 3 conventional trials in the literature. Each of those took about 8 years from the design to the publication of the results, 6 years of recruitment, because you do the patients and you need to follow them up for about 5 years. And for a study that has only about 108 patients in the end, the cost is somewhere around between 20 and 40 million pounds, and there is about 3 of them. This is one of the most studied devices. So the study that we published last year in Nature Communications was the first in silico trial on flow diversion devices. It took, and we compared the results with the 3 conventional trials in the literature. It took about us 3 months to run compared to the 8 years. It took us probably about 8 years to develop the models, but once you have the models, you can do as many trials as you wish with the same methodology. We had to develop a virtual population, compare that to to the populations of the 3 conventional trials before, to show that there was no statistical significant differences. And I'll save you the details from those, but the bottom line is we could reproduce the results of the 3 other trials to a 5% accuracy in a fraction of the time and the cost from those conventional trials. The advantage is that not only we can reproduce those, but we can understand aspects of those devices that are impossible to do through conventional trials, because it would be unethical or impractical to do some of the tests that you would sometimes want to do. Like, for instance, it's known that when aneurysms have a side branch, this side branch sometimes brings complications. And we managed to explain in that paper why that happens, which is something that there was only anecdotal evidence, but no actual trial that demonstrated that in the past. We could also do specific subcohort studies, so subgroups like patients with normotension and hypertension, which you couldn't do in a normal trial because the numbers are very small. And of course, you wouldn't be able to do a stress test ethically in one of these patients because of they're normally, quite ill. So to finalize, I want to say this is something which is picking up across every continent. And I'm just putting here 4 documents of Europe, the UK, China, and the US, showing that there is evidence of the need or explicit mention of the importance of in silico technologies, and that therefore we need as a community, and I hope that this association will help us to get there as well, to combine all the power of the science that we can bring together with the collaboration with the industry, working with regulators and with decision makers. And with that, I'd like to, I think I'm in my time, so I'd like to stop there and thank you for your attention.
Thank you very much, Professor Frangi, for your wonderful presentation, precisely on time, so it's really wonderful. And also the work is really amazing, and your call for collaboration. I just want to say if anybody have a question for Professor Frangi, I ask you to put it into the chat in the format of to the professor, to the presenter's name, put the name down. You can choose the surname, which is easier to identify. Put your name from, okay, your name, your email address. So if there's a communication and we know where to do that, answering to. And then put question there. So I put a format there. Unfortunately, the program is very packed, so we wouldn't be able to do a question and answering session now. So I want to thank Alex again for your wonderful presentation.
Professor Wan, perhaps what we could do, the speakers, is put our email as well, so people can contact us if they want as well. I'll do that.
That would be that would be really wonderful. Thank you very much, Alex, for the suggestion.